Hey, thank you, Todd. Thank you very much for coming on this beautiful Friday afternoon. Can everybody hear me okay? So just right now, before the seminar, Mark was telling me that I don't understand the title of your presentation. So, and a lot of times when I tell people that I'm working in toxicogenomics, they're like, what? So what basically it is, I'm studying effect of stressors, environmental stressors, on the genome of the organisms that they exposed to those stressors. But before I tell you about my update in the research, I want to tell you a little bit about myself, because a lot of times when I'm being asked where I'm from, I will tell I'm from Ukraine, but technically it's not correct. Actually, I was born on far east of Russia, near Vladivostok, actually right next to Japan Sea. And my dad was in military. He was in charge of building railroad, mainland railroad from Baikal to Amur. So basically all my childhood and middle school and high school, I was traveling from village to village, pretty small places. And we don't call it Siberia, we call it taiga. Siberia is before Baikal, so which also surprising to me when people is like, you're from Siberia. I'm like, no, taiga has a lot of coniferous and deciduous forest. So in 1992, we moved to Ukraine, my family, and we moved to Ukraine. It's western part of Ukraine. It's bordering Hungary. The name of the city is Uzhhorod. And 92 was the same year where perestroika happened within 6 months after we moved. And it was my first experience switching between languages overnight. Practically, we all switched to Ukrainian. So I went to Uzhhorod National University and got my BS in biology in 1995. And then after that, I proceeded to graduate school in Uzhhorod and got very interested in actually Chernobyl effects on plants, especially on flavonoid compounds in plants. And that's where I met my future advisor in Chernobyl. I volunteered to help American scientists with sampling in Chernobyl while I was there. And he is the one who actually offered me to come and apply for University of Georgia and become his student and continue working in area of population genetics, studying effects of radiation on plants from Chernobyl. And I'm forever thankful and grateful to Mark because he was just exemplary advisor, and I always feel thankful to him. And this is a picture of us where we both in Chernobyl processing our plants, which is kind of funny because I am all camouflage and protective clothing, so I don't get any soil contaminated on myself, wearing gloves. And Mike is like, sneakers, no gloves, no coat. It's like, whatever. So with that, after I graduated from University of Georgia, I went and did my postdoc at Savannah River Ecology Lab. And Savannah River Ecology Lab, it's in South Carolina on Savannah River site, that is Department of Energy site. And since 1950s, it was producing nuclear materials for nuclear weapons like plutonium. And I think it's the largest producer and the only producer of tritium in the United States. And now it's mainly the site of the waste storage and cleanup. So that's where I got interested also in population genetics, but in multi-generational effects of radiation. And as Todd already said, I came to UK in 2008 as scientist and transitioned to assistant research professor in July of 2011. So my DOE is mainly research, and I have 8% about teaching component. It includes also me being on committee of the students and plus teaching my class, Population Genetics Laboratory. It's a small class, but now you know why I'm teaching population genetics, because it's where my training was coming from originally. So my research accomplishments, while I was at UK, I have 43 publications, actually a total, but at UK 26 and 21 since I became a faculty. They're pretty highly cited. They're H-index 17, and I have a lot of citations. I actually surprised to find out just that the papers that we published with my PhD student, Daniel Starn, who graduated just last year, it's already been cited 60 times. So I guess we're in hot area of toxicogenomics. And from funding, so it's almost 5 million and 1,800,000 estimated to my program of toxicogenomics. And I have 2 PhD students. As I said, Daniel Starn graduated, and right now I have Anya Vamucha. And Anya, here he is. I was like, cannot find Anya. Anya is PhD student in the Department of Toxicology and Cancer Biology, and I'm on graduate faculty there. And here you can see me also with my summer undergraduate students, Summer Miller and Tyler. Tyler was through NIH SURES program. And also we have my postdoc, who was postdoc for the first 2 years, and then was Jason's postdoc. And I have Stuart, who started as Tracy Farmer internship before, but now he is a PhD student in our lab. So with that, I have a lot of projects that I've been working on during those years since 2011. And most of them involve actually examining effects, toxicogenomic, not genomic levels of nanomaterials in different organisms, plants and animals. I'm still keeping a little bit of my passion of population genetics and looking at evolution of plant breeding system, and it's wood sorrel, Oxalis alpina. That's why. So, and it's in collaboration with California Irvine Institute. So the project that I want to tell you about today will be actually a project examining effects of silver and zinc oxide nanoparticles, and also multi-generational effects of silver nanoparticles, all in the model that we are using, C. elegans. That's nematode, about 1 millimeter length. And no, Mark Peter did not contact about it. So I'm also a co-PI member of Center for Environmental Applications of Nanotechnology, which consortium of 7 different universities in US. Tiny transatlantic initiative for nanotechnology and the environment. We already finished this project, but that was in collaboration with United Kingdom people. And currently we also have collaboration with Nanofarm, which is fate and effect of agricultural relevant nanomaterials. And it's collaboration with Portugal, University of Aveiro. I wasn't sure if I need to put slide about what nanoparticles are, because you heard from our students, from Jason, but at least like very shortly, nanoparticles, they're just structures that are less than 100 nanometers in 1 dimension. And when you go on that scale between atomic and microscale, they result in very emergent novel properties. And this is just an example of one of them, like optical property changes. Like as the gold nanoparticles go from larger side to smaller side. This is TEM, transmission electron microscopy image of one of silver PVP coated nanoparticles that we've been working with. They're about 50 nanometers in diameter. And applications, wow, there is a lot of products that contain nanomaterials. This is just approximation. It's consumer product inventory, so the products that reported. And look, in 2017, we have about 2,500 products that just reported on the market. Application example of silver, they have antimicrobial properties, so you will find them in appliances and in clothes, in toys, in medical instruments. So they are being already used, nanomaterials containing products. So what happens with those products after residential use or after production? Those nanomaterials are being released and they are ending up in wastewater streams and go through wastewater treatment plant where they get partitioned to sewage sludge. And in US, actually 50% of sewage sludge is applied to agricultural soils as biosolids. So that's how they enter agricultural or terrestrial environment. And they further go by biochemical transformation and interact with a lot of ligands, can be taken by plants, can go to trophic transfer, and so on. So if you think about nanoparticles in the products and when they are released, I would call it as synthesized or pristine nanoparticles. Once they go into wastewater streams and partition to sludge, they constantly undergoing transformation. So this is a paper of one of our collaborators, Ruima, and they actually look at speciation of silver and zinc that were silver and zinc oxide nanoparticles. So wastewater treatment plant, pilot wastewater treatment plant, was receiving mixture of those nanoparticles, and they looked at actual speciation and the sludge. What they find out that the primary speciation product are zinc sulfide and zinc phosphate, as well as zinc complexes with irons, oxyirons. So major transformation process for silver nanoparticles was found to be sulfidation, and for zinc oxide nanoparticles were phosphatation and sulfidation. So now it brings me to the main questions that we want to ask in order to help with risk assessment of those nanomaterials. And the main questions, we work with metal nanomaterials that are prone to dissolution. So we actually want to know if the toxicity of those nanomaterials are as toxic as their counterative ions, basically. So if we have silver nanoparticles, are they as toxic as silver ions that being released from those nanoparticles? Also, as I mentioned about all transformations that nanoparticles undergo into, we are interested not only in pristine nanoparticles, but also is the toxicity, bioavailability, and all effects are the same between pristine and H particles or not. And finally, what I want to know is, can they cause multi-generational effects that eventually can be translated into population level effects too? So first I will start with actual studies that Daniel did. And this study is published in Environmental Pollution. And it's study on silver nanoparticles. Silver nanoparticles, MNP, I didn't mention, stands for manufactured nanoparticles. So it's always will be MNP because there are also natural nanoparticles exist. So he studied effects of pristine nanoparticles and aged nanoparticles, which were artificially aged in the lab, and they result in sulfidized nanoparticles. So when we look at the effects, first on toxicity, we look at mortality and reproduction on C. elegans. And again, they were exposed to silver ions, and we use silver nitrate, and pristine nanoparticles and sulfidized silver nanoparticles. So if you look at the scale, you can tell that exposure to silver ions results in the highest toxicity, followed by exposure to pristine silver nanoparticles and sulfidized silver nanoparticles. The same happened with reproduction. Silver ions are the most toxic, and aged nanoparticles or sulfidized were the least toxic. If you look at the scale, reproduction was the more sensitive endpoint. The difference between basically AC50 is concentration where you expect of decrease in reproduction 50%. And you can tell how much difference between AC50 for silver ions versus pristine and aged nanoparticles. So the bars that are shown in red is the actually estimated percent of mortality that would be due to dissolution of those particles at those concentrations. Again, it kind of concentration dependent, because at the lower concentration, we have almost complete dissolution. But at the higher concentrations, the mortality cannot be explained only by ions. And this is our dissolution data. And again, I want just to point out that those that dark, the ones that without pattern, are the ones that are sulfidized particles. So you can see the dissolution of sulfidized silver nanoparticles are much, much lower than the dissolution of pristine particles, right? And pristine particles, we have it over 3 hours and 24 hours. So another thing is that dissolution is concentration dependent. So there is a higher dissolution at lower concentrations than at higher concentrations. So what we also did, we wanted to look at distribution of silver inside of the nematodes. So we exposed nematodes to equitoxic concentration. So they weren't exposed to the same doses, but they caused the same effect. And it was 30% mortality in 4 hours. And we used synchrotron X-ray fluorescent microscopy at Argonne National Lab. And if you look at intensity of the silver, basically distribution inside of the nematodes, it's pretty amazing. You can tell that those nematodes exposed to sulfidized particles barely took any silver inside, but they still cause the same effect, the same mortality. While actually distribution of silver in ion exposed or in nematodes exposed to pristine particles was kind of similar. So the next thing what we did, we used actually a C30, so decrease in reproduction that caused 30%. And we continue to investigate transcriptomic effects. So for this, we use microarrays in our microarray facility. And after we got our data, first we did hierarchical clustering histogram that's shown right here. So I call it kind of like treatment independent hierarchical clustering histogram, because the replicates are grouped together independent of their treatment based on actual patterns of gene expression. And the one in red is positively charged, and the one in blue genes that are shown that, oh, what am I saying? The one in red are upregulated, and the one in blue color genes are downregulated. Sorry. So you can see that actually replicates did cluster together within their respective treatments. So if we, for example, expected effects just due to ions, you would see almost random clustering of those replicates. But we also look at the Venn diagram, right? And this is number of genes that are unique to each treatment, and this is the number of genes that are in overlap, so they're shared among treatments. So if you look at that number, there was only 20 genes that were shared among all treatments. And if you look at the number of genes shared between silver ions and pristine, we had 54 genes, and one of the genes was nuclear metal responsive gene, which actually responds to ions, which would be indicative of effects due to ions, at least in pristine nanoparticles. And I didn't show those data, but we did confirm it with using mutants, and mutants were more sensitive in response to pristine and ions than wild type, confirming that the gene does involve in the toxicity. So among the 20 shared genes, another thing that was interesting, there were almost 1, 2, 3, 4, 5. So 11 genes that responded completely in opposite direction. So even they were, among shared genes, they weren't responding in the same way. And they were only responding in different way by upregulation in response to sulfidized H silver nanoparticles. But they were downregulated in response to ions or pristine particles. So those genes are called ground-like genes. They're kind of related to hedgehog gene family. and there are some evidences for those genes to be involved in cuticle formation. And another 6 genes were collagen genes, and also involved obviously in cuticle structure and function. And C. elegans has a lot more than 100 genes. I think it's 141 gene of collagens. Okay. So the next thing that we did, we actually decided to investigate biological pathways that would be induced to those treatments. And the first thing, again, it's Venn diagram that you want to see. There is nothing in the shared area. None of the pathway was common for responding to all those treatments. So they all had distinct pathways, further indicating that probably not all the responses are due to silver ions and dissolution of silver. And interesting that, for example, pristine nanoparticles, one of the pathways was lysosome pathway. And lysosome is basically, I call it waste disposal factory that can dispose debris, molecules from inside the cells and outside the cells. And some of the lysosome proteins that were upregulated were cysteine proteases genes that are responsible for proper protein degradation. Another thing that's important for us to know that lysosome has a very acidic environment. So we know once nanoparticles get into lysosomes, they will be dissolved in those conditions and silver ions will be released. In terms of silver ions, we had actually the genes that responded to, for example, catalysis genes. So that would you expect in response to reactive oxygen species like hydrogen peroxide. They were all upregulated, which is not surprising, and you would expect it in response to silver nitrate, silver ions. And another series of genes was actually glutathione S transferases genes. And those are the genes that involve in detoxification phase, second phase of detoxification. And it's responsible for conjugation reactions for reduced glutathione to xenobiotic substrates. For our H, sulfadisilver nanoparticles, the only pathways that was induced was calcium signaling pathways. And this nematodes is interesting that, for example, if you want to relate calcium signaling to reproduction, then it's hermaphrodites. So when oocyte is entering spermatheca, there is a huge flux and increase in calcium that promotes actually that fertilization, meiosis, and eggshell formation. So it wouldn't be surprising to see calcium signaling. But also an egg 30 protein, it's egg laying deficient protein. And UNC genes, it's called uncoordinated genes. Mutations in those genes usually associated with sluggish, kind of paralyzed, slow moving phenotype. And a lot of times, and especially those are present in muscles. So conclusion for this silver nanoparticle study, what did we find? So just to summarize it. So we saw that sulfidized particles are less toxic and less bioavailable, and their dissolution is also decreased, right? Very low dissolution. Some of the pristine nanoparticles, their toxicity was due to silver ions, and we did confirm it with using mutants. And I did already said that at the same time, all treatment produce distinct pathway, indicating that there is more to it than just toxicity due to ions, that some of the effects are particle specific. And you saw that taken together with bioavailability data, where sulfidized particles barely take silver inside, and there seems to be their responses, toxicity responses, are due to damage to cuticle proteins more than anything else. So it's again the study that was done by Dan. And now I want to switch focus a little bit to zinc oxide nanoparticles. It will be in the same plan. Again, we are looking at toxicity and genomic responses, but this time we have actually 3 different treatments, 4 including controls. So we have pristine zinc oxide nanoparticles, then 2 different H zinc oxide nanoparticles, phosphate H and sulfidized. And also, of course, we included the zinc ions in the treatment. So this is TM images, oops, of all those particles. And first of all, I wanted to look at the dissolution. And if you look at the dissolution of pristine zinc oxide particles, and this is in synthetic soil pure water. So they basically completely dissolved because they're not much different from zinc ions. But among the aged ones, at lower concentrations, the dissolution is higher for phosphate H, lower for sulfidized. But once we get to the concentration, higher concentration, and it's the concentrations that will be corresponding to a C30, the dissolution is still exactly the same for phosphate H and sulfidized. So when we look at mortality and reproduction, it's a little bit different than silver. So it was hard to represent it, but I want to point out that if you look at the scale for ions and for pristine zinc oxide nanoparticles, it's exactly the same. So you basically would overlap those pristine over ions. So the responses are very similar in mortality to ions and to pristine. With H particles, again, the scale and this scale, it's exactly the same. So the response is very similar between H, but obviously they're show not that much toxicity in comparison with ions. But honestly, overall, the toxicity in zinc oxide study was much less than in silver study. We actually, mortality-wise, we could barely reach 25%, and we went pretty high. So with reproduction, again, more sensitive endpoint, the same story. No difference much in effect of reproduction between ions and pristine particles, and no difference between 2 aged, but H is significantly less toxic than ions or pristine. So we were interested in speciation of zinc inside of the nematodes, and we used X-ray absorption near H spectroscopy to do it. Everything that you need to know here is that basically this is spectra for model compounds first. This is spectra for our treatments for nematodes exposed to ions, pristine. The one that was phosphate H, they were phosphate H at pH 8, so that's in green that correspond to that, and control. So the best fit of actually of those spectra was fitting to acetate, zinc acetate and zinc glutathione. It doesn't mean necessarily that exactly the compounds that were only forming inside of the nematodes, but it means that zinc was actually forming complexes with sulfhydrals and carbohydrals. So there is no zinc oxide or zinc phosphate was found in the nematode, only zinc ions. Okay, if you saw the previous Venn diagram was a little bit complicated, this one, it can get really interesting when you start playing with transcriptomics. Just to walk you through, the main thing we are looking again, how many genes respond to ion treatment, 147. How many genes were shared among all treatments, 79. So out of the genes that responded to ion treatment, 54% of those genes were shared among all treatments. And interesting that among these genes were, again, nuclear metal responsive gene and 2 metallothionein. And metallothionein are obviously responsible, it's cysteine-rich proteins and responsible for metal detoxification as well for zinc homeostasis too. They both upregulated. So further indicating that definitely this is some effect due to zinc ions. Looking at biological pathways, wow, that's a lot of kind of information, but I was surprised to see how many pathways were activated in response to phosphate H, zinc nanoparticles. But among shared pathways that I want to look at again was lysosome and ABC transporters. Lysosome pathway was shared basically among all aged and among ions. But also when I looked at pristine zinc oxide nanoparticle treatment, it just cut off that we did 1.5-fold change. So if you look in response to pristine zinc oxide nanoparticle treatments, again, they had lysosome genes that were still responded at 1.4-fold change. So even though it doesn't show up as shared pathway, I would still think that there are evidences for lysosome pathway activated in response to pristine too. And ABC transporters, it's basically, it's also involved in metal detoxification, and there are interesting proteins. It's big P glycoproteins, BGP2 and 5, and I just want to point out, I think it's BGP2. That BGP2 protein is involved for gene that coded for that protein. Information of zinc granules that forming in the site of the C. elegans intestine. And when there is zinc, excess of zinc, they're actually storing that zinc in those granules. That's similar to lysosomes, but much bigger. That gene also responsible for morphological change of those granules in two different compartments. One that's acidic and one is not, which would form a storage, and one that would prone to dissolution, that would result in dissolution of those particles, and the others that would just store those particles if they get there. So in conclusion for this study, for zinc oxide, and to compare it to silver, we obviously saw some differences. So we saw that in contrast to silver study, ions weren't more toxic than pristine in zinc oxide pristine particles, right? So zinc oxide particles were as toxic as ions, and we didn't see it in silver. But similar to silver, again, H particles cause much less toxicity than pristine particles. Based on speciation data, we found that only zinc ions in different complexes present inside of the nematodes, suggesting that we see a lot of effects due to dissolution. And also that higher number of genes that we detected as shared among all treatments, and plus the genes that respond to metal ions further confirms that a lot of those toxicity due to ions. So overall, the biggest conclusion of that there is much more evidence for zinc study that actually toxicity is due to ion released than, for example, in silver study. I didn't mention, guys, if you have any questions, because right now I'm going to shift gears toward actually our final study that I want you a little bit to tell about. And it's study done by Anya and Caroline first. And Caroline actually was a student who visited here from Oxford, and I was basically US advisor for her dissertation work. And what Caroline did, she looked at multi-generational effects together with Anya. What they set up here is basically exposures over multiple generations, over 10 generations. So the nematodes were exposed over 10 generations to, again, ions, pristine sulfidized particles, exactly the same particles and ions that we are using in our study. And also there was recovery population. When after 5 generations of exposure, the exposure was stopped and then let it recover for 5 generations. So that would be recovery population. At different generations, what Caroline did, looked at those response relationship in response to reproduction, basically. So what we found was interesting, that there was significant actually decrease and increase in sensitivity. Look, this is AC50 values. AC50 meaning concentrations that correspond to decrease in reproduction 50%. So they dropped almost right away as early as F2 generation, and they continue at that level up to F10 generation in response to ions and pristine silver particles, but not to sulfidized particles. Also, these open square represent your recovery populations. So the recovery populations did not recover, even if exposure was stopped after 5 generations. So what's happening? Why there is such increased sensitivity in reproduction? And Anya, you told me I can use any picture, right? So I thought that would be a great picture to use, really like it. So Anya is the one who actually asking question if exposure to those silver nanoparticles can induce genomic instability, can overall increase mutation rates in C. elegans. And I put the design of the study here just to give you an idea of what he did. Basically, we did expose also C. elegans over multiple generations, 10 generations, but we followed single lineages for each replicate. And then he collected offsprings from every generation. But the ones that we actually sequence and use next generation sequencing and DNA-seq method was from generation F0 and generation F10. The nematodes were all exposed at AC30. So again, different concentration, but cause the same level of effect. After we did 4 rounds of deep sequencing with NextSeq, and we got to 16x depths in general. And after lining our reference genome and using quality filtering and filtering as many variants as we could to avoid actually false negatives and false positives, right? We end up with those results right now, and I just want to say that it's not final results. We are still processing and trying to see if we need to remove pre-existing SNPs. That when we met with Mark recommended actually that we do need to remove the single nucleotide polymorphisms that are due to parology. And when you blast actually genome against itself, you can find those SNPs that usually already mutated. So after removing all those, you can tell that there is maybe pattern toward increase in the number of homozygous mutations where both copies of gene are mutated. In response to pristine particles and sulfidized particles, again, they're not significant, but there is some pattern, something going on. And those just total number of mutations, those single nucleotide polymorphism, deletion, insertions. If you look at heterozygous mutations, it's many, many more. We don't see any differences among treatments, not even pattern. So definitely, as I said, still being analyzed. But can this finding explain what we were trying to actually explain and why we started the whole investigation of this study? So we were trying to see if we can find explanation why there is significant decrease in reproduction in F2 as early as F2, why they're more sensitive to ions and pristine. And if you look what we found actually increase in homozygous in response to pristine and sulfidized, but not even ions. So it definitely does not explain the results that we saw before. So what it pushed us to go and look that maybe we are not looking in the right place. And it's not due to necessary germline instability and increase in mutation, but epigenetic mechanism. And what epigenetic really is changes, heritable changes in gene expression levels, but it's without any actual like physical change in DNA sequence. And here I put just two mechanisms, in DNA methylation and histone methylation. And basically we started with DNA methylation first. DNA methylation, so what happens when methyl group attaches to DNA, it kind of can silence the gene is not expressed. And this is also can be transferred to offsprings. So with C. elegans until 2014, it was believed that the genome is not methylated at all. In fact, I saw the data that is like 0.0002% methylation in the genome at cytosine level. And then Greer et al., they discovered the genome is actually methylated at adenine. And you can use this methylation to investigate the changes in total global level of methylation in C. elegans in response to those particles. And I use slut bloods, and obviously with antibodies specific for adenine. And what we found, well, if you look at those results, right, this is the DNA methylation level relative to controls, and those are all our treatments. What I want to point here is F0 generation, F2, and F4. F0 is non-exposed, F2 is exposed, F4 is recovery. So basically, it looks like there is decrease in level of methylations in response to pristine in F2, and then it stays not recovered. But be very careful, because if you looked at what happens at F0s, which weren't exposed, there is a lot of variation going on there. And it's not really clear, there is significant variation. So something is going on with changes in DNA methylation, but we definitely need to look further before we really say that it does cause decrease in DNA methylation in response to pristine and increase in response to ions. So what's next? We actually want to use liquid chromatography, mass spectrometry to try to figure out the actually methylated adenine. It's like more precise method to measure global DNA methylation. And after listening to Sharon's talk last week, I realized that we can use, and we talked with Anya and discuss it too, that we can use CHIP method to pull the DNA that is methylated and sequence it and get actually to the genes that may be related to reproduction. Also, we have in plans to examine histone modification, mainly methylations first. And our projects that we are already started working on, and Anya and Tyler both involved in that project, is with Nanopharm, looking at the toxicogenomic effects again and multi-generational effects of copper hydroxide nanoparticles and the agricultural fungicide, coside that contains those particles. So with that, I would like to thank everyone who came today and everyone in our lab. It's a lot of people, a lot of students, postdoc, former and current, a lot of my collaborations. Some collaborators are obviously in United Kingdom and University of Portugal and all over US. People who helped me with running my course that I'm teaching, population genetics lab course, and so on, and obviously my funding sources. And my family. Jason, you were on that slide, but he's more just collaborator, obviously. He's the one who got me interested in Nana, frankly. And he's great. He's great dad, and love our kids. And when I showed him picture of Stella, he said, wow, live long and prosper. It's a sign. And I'm like, wow. So live long and prosper, smile often. And now I would like to take any questions.
Seminar, how worried should we be about human health after all that you found these studies?
That's questions that won't be that easy to answer, but from what we studied, right, you can tell that the H nanoparticles, and we probably are being exposed just to H nanoparticles that are transformed, they are definitely less toxic. So if we can regulate them on at least ion level, that maybe we're okay, but I wouldn't say for sure, because we don't know a lot of effects. For example, some studies showed that some nanoparticles can be dissolved, like when they're exposed to exudates of plants, right? So we still need to be really careful about how we regulate those particles.
Is there need no regulation on the waste treatment level?
Not that I know of. Jason, do you?
No.
No, I mean, I don't think, yeah, I don't think so.
How much do plants take up particles and how do plants transform?
Depends on the plants, actually. Some plants can take quite a bit. The first study that ever been confirmed actually in Savannah River Ecology Lab was plants were tomato plants that were taking quite a bit of gold nanoparticles, and they can translocate it to the leaves, for example, too. The fruit, I think some plants take only in the roots. I'm trying to remember the study that was done actually on the fruit, and I don't remember them detecting that much of nanoparticles present in the fruit. Yep.
So, about the genetics, is there any question on how to the question is, do you see any differences in this modification of this pattern?
Mm-hmm. There is definitely, actually, C. elegans is very conserved if you compare it to mammalians, and there are key that you can look at 21 modifications, and I think like 80% of those modifications are present in C. elegans. But I think it's histone methylation mainly that was shown to relate to reproduction. So, histone variants? No, it's H3K9. Oh, I see, I see. That's actually a good point. Yeah, we will look into it. It may actually give you, see the transcriptome data. I mean, talking about hairballs, right? So you think you'll take those transcriptome data, you know, and there's beautiful pathways, but then you end up with one or two pathways and a lot of random genes. And actually not that many, I don't remember seeing that many histones. In particular, I was looking at histones and I didn't see them. But we need to go back and look at it a little bit more carefully again. But yeah, it's a good point. Yeah. No, it's a good point. I mean, if we ever get to that, because when we look at the price of that kit, we immediately decrease the number of modifications that we're going to look at. But
Within multi-generational studies, what was the typical sample size for different generations? How many individuals with C. elegans were you looking at?
Oh, with C. elegans, you cannot look at individuals. So basically what Anya does, he extracts the whole progeny. So you start with individual C. elegans,
Okay.
but then you let it reproduce. And then you get DNA from the whole progeny. That's how, and then you take another part and continue it again, C. elegans and continue. So I would say it's about up to 100 nematodes. Yeah, but we used 4 replicates for the sequence and DNA-6 study.
It looked like the shape of the distribution didn't vary with the generation at all in the sort of cartoon version that you showed. Is that the case?
Wait, are you talking about the...
From the F2 to the F10?
Yeah, from those response studies for AC50? Oh, you're talking about that. I'm talking about DNA-6. So the shape of response, I was just showing actually how AC50 changes in each generation. So it wasn't the whole dose response, but they are in the paper too. I mean, the distribution, I don't think they changed that much, but the fact that AC50 was keep decreasing. Right, in F2, but then it stayed about the same level. Okay. Any more questions? Mark?
Is sulfide itself directly toxic?
No. No, actually, it's not, and we have a lot of it present in our media. So it's controls them just great. The amount of sulfides that present in H particles wouldn't be as much as we already have in the media. So the same with phosphate.
So the general impression I get from this is that the nanomaterials that you studied so far are not more toxic than they're toxic.
The toxicity mechanism and maybe where they're going once they're being taken in and if they're dissolving inside. Or... Then free ions, yes, they were. Yeah, that's very good news. Yeah, and once they transform, they're even less toxic. So that's even better news.
So the worry is
we didn't see it. Yeah.
Well, thanks everyone for attending. Before we leave, I do want to invite faculty of Clinical Sciences to stay afterwards for a discussion of older promotion dossier. Thanks everyone for attending, and let's thank Olga for our presentation.
Gosh, that's a lot. That's a lot of microphones.